Welcome to today's lesson on the development of our understanding of genetics, where we will delve into the pivotal work of Gregor Mendel and the remarkable journey of his discoveries from obscurity to key principles in modern genetics. In today's lesson, we will explore two main objectives. First, developing our understanding of genetics through the lens of Mendel's pioneering experiments, and second, understanding why the importance of Mendel's discoveries was not fully recognized until after his death. So far, you have learned about genetic variation and evolution, key concepts that lay the groundwork for understanding how traits are passed down and how species evolve over time. This foundational knowledge sets the stage for us to introduce the key milestones in the history of genetics that explain how we have arrived at our current understanding. By exploring Mendel's work, we gain crucial insights into the process of scientific discovery and validation. Understanding these concepts advances our biological understanding, highlighting the importance of observing, experimenting, and critically analyzing data over time in the scientific community. Let's get started. Gregor Mendel, an Austrian monk with training in mathematics and natural history, conducted pioneering experiments on plant heredity in the mid-19th century. Working in the garden of his monastery, Mendel studied how various characteristics in pea plants were passed down from one generation to the next. His research, published in 1866, laid the foundation for what we now know as genetics. Firstly, he discovered hereditary units. Mendel proposed that characteristics in plants are determined by hereditary units, which we now call genes. Second, he suggested that these units are passed on unchanged from both parents. Each parent contributes one unit for each characteristic. Third, Mendel suggested that some hereditary units are dominant, meaning they will be expressed in the plant's appearance if present. Others are recessive and will only be expressed if the dominant unit is not present. For example, Mendel found that the unit for tall plants, capital T, was dominant over the unit for dwarf plants, lowercase t. Therefore, if a plant inherits the unit for tallness from one parent and the unit for dwarfness from the other, the plant will be tall. He also found that if these tall offspring were crossed in another generation, a small portion, about a quarter of the offspring, will have dwarfness. This is because the recessive small t alleles are carried by the offspring and can be passed on to the next generation without being displayed in the phenotype. While Mendel's findings were revolutionary, they were not recognized by the scientific community during his lifetime. There are a few reasons for this. Firstly, the lack of fundamental knowledge. At the time, the concepts of genes, DNA, and chromosomes had not been discovered. Scientists lacked the context needed to understand the significance of Mendel's work. Secondly, limited communication. Mendel published his findings in an obscure journal with limited distribution. As a result, his work did not reach a broad audience. It wasn't until after Mendel's death that other scientists, with a better understanding of cellular processes and inheritance, began to recognize the importance of his research. Gregor Mendel's pioneering work set the stage for future scientific discoveries in genetics. Key developments that followed include chromosome observation, the chromosome-gene connection, and the discovery of DNA structure. In the late 1800s, scientists observed chromosomes during cell division and noted their behavior. By the early 20th century, it was proposed that Mendel's hereditary units were located on chromosomes. These units are what we now refer to as genes. In 1953, the structure of DNA was determined, which significantly advanced our understanding of gene function and the development of gene theory. These advancements confirm the profound impact of Mendel's work on our current understanding of genetics. In today's lesson, we explored fascinating history and development of genetics, with a particular focus on the pioneering work of Gregor Mendel. We began by examining Mendel's groundbreaking experiments with pea plants, where he formulated the fundamental laws of inheritance, including the concepts of units of hereditary and dominant and recessive traits, and observed trait inheritance patterns. Mendel's work laid the foundation for our understanding of genetics. Following our discussion of Mendel's work, we learned why his contributions were not immediately recognized during his lifetime. We noted that during the 19th century, the scientific community lacked the knowledge of DNA and was not ready to integrate Mendel's findings into the broader understanding of biology. It was only after the rediscovery of his work in the early 20th century, coinciding with advancements in microscopy and the study of chromosomes, that the significance of his discoveries was fully appreciated. Through this lesson, we highlighted the importance of Mendel's work as the cornerstone of classical genetics and its enduring impact on the field. We learned how his principles are not only fundamental to our current understanding of genetic inheritance, but also paved the way for later discoveries in molecular genetics.